Well, I'd like to start by thanking the organising committee for this very sort of kind invitation to come and talk about some of our recent work that we've been doing in Birmingham. I was sort of reading this sort of abstract book and I noticed the chair sort of referred to me as a sort of towering presence in the mycobacterial field. I'm sure he wasn't referring to my research, but more to my height as an Asian, being very tall. I was going to talk about the cricket a little bit, but I thought that would be a bit too much. So why don't we just start with the science? So what I'd like to do today is give a sort of brief introduction about some of the problems that we face with tuberculosis, and then leads into some of the research that we're doing. So really, tuberculosis has been around for many, many centuries. And I wanted to point out a couple of articles here. This one in the Proceedings of the Biological Society run by the Royal Society, where you can actually take sort of skeletal remains and actually analyze them for the presence of tuberculosis. And here we're using sort of chemical methods to detect chemical markers, such as mycolic acids. More recently, I guess if this meeting was probably a couple of weeks ago, this would have been hot off the press. A publication in collaboration with Stuart's Cole group in Science, where we've looked at a comparison of sort of modern and medieval strains of Mycobacterium leprae, which is related to tuberculosis. In terms of Mycobacterium tuberculosis, it's really a sort of complex of organisms and all interrelated. So you have Mycobacterium tuberculosis, which affects humans. Then you also have Mycobacterium bovis, which affects cattle, which is quite a huge problem in the UK, as if you've been reading the newspapers in terms of people have been advocating the culling of badgers. I guess you have a similar problem here with possums. But really, there's a sort of evolutionary bottleneck, and this is probably an underestimation, but these have probably arisen through a single sort of Mycobacterium species. So in terms of a simplistic sort of form, obviously we have ourselves as the host. These are the bacilli residing in a macrophage. This is what they look like on solid agar plates. Obviously, this is the infection state. What you tend to get is an immune response. Either this will go on to lead to disease, and this is the characteristic sort of chest X-ray profile that you get with a tuberculosis sufferer, or you get immunity. I've got here reactivation in that you can go via this pathway, in that it's sort of estimated that a third of the world's population is in fact infected with Mycobacterium tuberculosis latently. So here you can actually carry the Mycobacterium tuberculosis bacilli, but actually not see any of the symptoms of the disease. In terms of case rates, there's around 8 million new cases of tuberculosis and 2 million deaths. And this shows the sort of geographic spread of the disease. It's really something that we find associated with South America, Africa, and parts of Asia. But the big problem we have is multi-drug resistant tuberculosis, which is referred to as MDR-TB, and also what we refer to now as extensively drug-resistant tuberculosis, XDR-TB. And these sort of map onto the same regions in terms of South America, parts of Africa, and Asia. So what are the main problems? And so it's this epidemiology sort of increasing and high sort of caseloads of tuberculosis really are associated to a number of things. One of them is really, if you look at geographically where tuberculosis is found, it's really associated with poverty and poor healthcare systems. The second sort of major factor in terms of tuberculosis incidence is its sort of co-infection with HIV. And this sort of highlights the sort of number of clinical cases you obtain with TB when you have sort of endemic populations with HIV endemic populations of tuberculosis sufferers. So what can we do? There are drugs available to treat tuberculosis, but they're quite protracted in terms of chemotherapy. You have to take 4 drugs initially for 2 months, and these consist of rifampicin, isoniazid, ethambutol, pyrazinamide, and then later for another 4 months, rifampicin and isoniazid is continued. I think one of the key things to note here is the discovery of these antibiotics. They all come from that sort of golden era of chemotherapeutics in the 50s and 60s, which I'll come back to later in my talk in one of the later workshops. If you look at the sort of mode of action of these inhibitors, most of the target aspects are cell wall synthesis, and this is where sort of my interest lies in terms of tuberculosis. So really, what are we looking for in terms of the problems that we find with tuberculosis? We're looking for new targets and the generation of new anti-TB drugs, a better understanding of tuberculosis pathogenesis, and also vaccines. So what I'd like to do is sort of show a sort of schematic representation of the Mycobacterium tuberculosis cell wall. It has this sort of underlying peptidoglycan, attached to which, through a sort of phosphate linked to the muramic acid of peptidoglycan, we have this unique diglycosyl bridge, which consists of rhamnose and N-acetylglucosamine. Attached to the rhamnose in red or orange, depending on what your color is here, you have a galactan polymer. Galactose is unusual in this particular organism that's in the furanose ring form, and that gives its unique specificity to the organism. The galactose residues are linked 1,5,1,6 in an alternating fashion. Attached to the galactan backbone here, we have these 3 arabinose stubs. Again, the arabinose here is unusual that it's in the furanose ring form, and it's made of quite distinct structural motifs in terms of through different linkages, whether they're beta 1,2, alpha 1,3, or alpha 1,5. Attached to the termini of these arabinose residues, we have what's really the most sort of characteristic property of Mycobacterium tuberculosis, the so-called long-chain fatty acids, which we term mycolic acids. These are basically sort of alpha-alkyl, beta-hydroxy fatty acids, and really the mirror chain, which is the longer chain here, is around sort of 56 carbon atoms in size. And the overall size of the fatty acids are around sort of C78 to C90. So they're very hydrophobic. And this is really the sort of hydrophobic layer that prevents the sort of penetration of conventional antibiotics through the cell wall. Intercalated within this sort of mycolic acid leaflet are a whole variety of unusual waxes and lipids that we associate not only with Mycobacterium tuberculosis, but with other genera of mycobacteria. We have the thiaceryl dimycosyrosates here. We have diacyl trihaloses, and a sort of derivative of the thiaceryl dimycosyrosates is the so-called phenolic glycolipid, where we have a sort of terminal phenol group esterified with a rhamnose residue here. In addition, we have a second molecule, which we refer to as lipoarabinomannan. It has a phosphatidylinositol anchor core. Linked to it, we have a mannan backbone, and this is sort of branched. It has a linear 1,6 backbone, but it has punctuated by alpha 1,2 branches. Attached to this, we have a similar sort of arabinan segment, and then we have various sort of capping motifs, which I'll talk about later. But in Mycobacterium tuberculosis, these tend to be mannose capped. So really, one of the challenges for us was to try to unravel the biosynthesis of these glycoconjugates. And this is quite challenging for a number of reasons. A lot of the enzymes are involved in the synthesis of these 2 molecules tend to belong to a family of glycosyltransferases, which are referred to as the GTC glycosyltransferase families. And these tend to use lipid-linked donor substrates and tend to be membrane proteins. In addition, a lot of these enzymes are essential in tuberculosis. So in terms of generating conventional mutants, it's very difficult to study the physiology of the Mycobacterium tuberculosis cell wall in a sort of conventional manner. But it's key in that we do know that these entities are targets of frontline antituberculosis drugs such as isoniazid, which targets aspects of glycolic acid synthesis, and ethambutol, which targets aspects of arabinan synthesis, both in lipoarabinomannan and also in arabinogalactan. So what is our sort of current understanding of arabinogalactan biosynthesis, or referred to as AG biosynthesis? So really, what we wanted to do is we wanted to develop a system where we could shuffle between different organisms. And I think this will become obvious as I start to talk about these different organisms and what we've been able to do. The three organisms that I've highlighted are obviously Mycobacterium tuberculosis, Mycobacterium smegmatis, and Corynebacteria glutamicum. And these all belong to the Corynebacteriaceae family. So they're all related in structure in terms of they have this common sort of mycelial arabinogalactan peptidoglycan complex that we referred to. And these have sort of various sort of useful properties. In terms of Corynebacteria glutamicum, it's possible to generate sort of deletion mutants which are actually stable, and this allows you to study the particular impact of a gene deletion within a pathway in a stable environment, which would otherwise be unable to do in Mycobacterium tuberculosis. Depending on the pathway, you can also knock out particular genes in Mycobacterium smegmatis and also study some of these glycoconjugates. So really, sort of shuffling between these organisms, we can generate a panel of deletion mutants and also conditional mutants to study both of these glycoconjugates. We also had to develop a number of key assays that were specific to these enzymes, which relied the production of recombinant proteins, where this particular organism can be very useful. So I mentioned the GTC glycosyltransferase family, but actually the biosynthesis starts via the utilization of the GTAB glycosyltransferase, RFE here. And basically these enzymes use sugar nucleotide donors. So here we're having the transfer of N-acetylglucosamine to this sort of lipid phosphate carrier, which is anchored in the membrane, to give our first intermediate. We then have the addition of rhamnose from TDP rhamnose by wobble to give this intermediate. And then we have the addition of two galactose residues, both in the furanose ring form, coming from UDP galactofuranose via this enzyme. And then we have a second galactosyltransferase, which actually zips this along to around about 30 residues in size. We don't understand exactly what dictates why the first enzyme is only extending 2 residues and the second enzyme extends up to about 30 residues. Both of them have a similar sort of molecular architecture when we look at the proteins. So that's a sort of unknown at the moment. At this point, this intermediate is then flipped to the outside. And we then start to utilize this sugar donor, which is DPA, decaprenyl phosphoarabinose. It has this decaprenyl moiety, has a phosphate, has this arabinose in the furanose ring form, and it's basically synthesized from ribose phosphate pyrophosphate here through a number of steps, which I'll talk about later. So really, what we were able to do in Corynebacteria glutamicum was to knock out this gene AFTA, and this provided us with a backbone which consisted of this polymer in that particular mutant. We were able to permethylate the cell wall, isolate this fragment, and using fast atom bombardment mass spectrometry, actually sequence the glycan chain and the precise position of the arabinose residues. And these turned out to be on the 8th, 10th, and 12th position of the galactan backbone. Some work that we'd done earlier while I was at Colorado State University with Patrick Bennon and Julia Innermean was to look at sort of ethambutol resistance and how we could mediate a thiambutyl resistance using a sort of cosmid library in Mycobacterium avium. And this really led to the discovery of the M cluster, and in particular M-A and B, which we hypothesized at the time would be responsible for the synthesis of the additional arabinan segments of arabinogalactan. I want to go back a little bit and talk about DPA in more detail, because it's quite a key sort of lipid intermediate and donor in terms of arabinogalactan synthesis. It starts, as I mentioned earlier, from pRpp, and we have the addition of the decaprenyl phosphate moiety catalyzed by UBIA here. And really, the key steps are here, DPRE1 and DPRE2. What we're doing here is we're having an epimerization reaction where we're changing the orientation of this hydroxyl on the 2 position, where it's down to up. So this would be ribose, this would be arabinose, and it goes through this keto intermediate. Using some sort of old-fashioned sort of biochemical techniques and generation of membranes and addition of appropriate substrates, you can actually follow the synthesis of this pathway through pRpp and look at each of the distinct products that are synthesized. So you can look at the formation of both DPA, DPR, and DPX, and also the phosphorylated form of DPPR. So this is sort of quite useful in that we established a lot of the basic biochemical assays at the time, and it became that this was a key enzyme in terms of the pathway, not only just for arabinogalactan synthesis, but also for lipoarabinomannan synthesis. So Stuart Cole, a few years ago, described a key sort of finding in that he discovered a range of compounds which referred to as BTZ, and I think it was Christophe Guyot who, in association with Mary Jackson, discovered these DNB compounds. So these are sort of highly active against DPRE1, and they have quite high potency against Mycobacterium tuberculosis. So these came through a sort of screening program that they were running at the time, and basically what they found was that resistant mutants had this sort of mutation at the active site. One of the things to note about these compounds is that they're quite hideous in terms of drug discovery. You know, one of the big things is this nitro group is a big no-no from what medicinal chemists tell me. But as a lead, they're sort of quite useful. So we went on to study some of these compounds, and what I want to sort of highlight here was that, you know, one of the key things is, you know, can you obtain recombinant protein expression? And we stumbled across this sort of technique due to one of my colleagues at Birmingham who has a long-standing interest in chaperone proteins. And we found that if you were to take M. tuberculosis proteins in E. coli in a classical sort of PET system and use a variety of E. coli sort of chaperones, you can actually facilitate folding and purification and high-yield, high-quality protein. So to go through this slide a little bit, so we were able to basically take radio-labeled substrate here, DPR, add in DPRE1, DPRE2, and basically look at the conversion of DPR to DPA. There's a little bit of the intermediate DPX here. What you find is that when you start to add in the inhibitors, so this is related to the BTZ compound, what you find is that you block the conversion to DPA and you get DPR. Likewise with BTZ. So this is quite useful in that they'd utilize this sort of phenotypic screening assay to identify these hits against this enzyme, but this had now proven that the inhibitors worked at an in vitro level. We went on more recently to solve the crystal structure. This was done with a colleague of mine at Birmingham, Klaus Futterer, and really it's been published recently in PNAS. It has basically the substrate in there, and also we've been able to get co-crystals of CT325, and also we have co-crystals of the BTZ compounds, and this shows the sort of network contacts of the different amino acids. So going back to sort of arabinan synthesis for arabinogalactan, we were able to show, again, using Corynebacteria, the role of this enzyme, AFTB, and basically it was involved in terminating the sort of synthesis of the arabinan segment. But what was missing at the time was how are these branches introduced, these 3,5 linkages. So what I was going to do was run through a sort of the techniques that we use in terms of how we try to identify different glycosyltransferases in Mycobacterium tuberculosis. And basically, you can go through and, using bioinformatics, you can do a pileup of all the different GTC glycosyltransferases, and you end up sort of various sort of plots like this for each one, and they tend to have these characteristic sort of motifs that you predict to be involved in catalysis. One of the things is that when you start to do pileups with related organisms, you can find a sort of high degree of similarity. So I'm going to talk a little bit about this gene, RV2673, at the time. So we identified that it was in Corynebacteria, also in Smegmatis. It looked like a GTC. So using a sort of method developed by Bill Jacobs at Albert Einstein and currently used by Dr. Apurva Pat in the lab, we were able to generate a deletion mutant in Mycobacterium smegmatis using his phage technology. And the mutant was stable, which was a little bit to our surprise. We thought that it would have a lethal phenotype, but it didn't. So that's the beauty of sort of checking whether you can actually obtain mutants across the different species. But what was interesting, that when you looked at the colony morphology, you had this sort of cross-granular morphology that you associate with Mycobacterium smegmatis, but you had this sort of slimy sort of morphology in the mutant. The other interesting thing was its sensitivity to drugs. What we found was that it became sort of hypersensitive to drugs, which sort of suggested that we had altered some aspect of its cell wall. This became more obvious when we started to look at its mycolic acid composition. So here in the mutant, you can see these are the classical sort of profiles that we run on thin layer chromatograms of the different types of mycolic acids. And here you can find that there's a very sort of diminished decrease in terms of the mycolic acids that we find on the cell wall in the mutant. Obviously, if we come back and we complement the mutant with both the smegmatis or the TB gene, we get restoration of full mycolate synthesis in the mutant. The other thing to note was that the other thing that we find is that in the extractable lipids, we find a lot of trihalose dimycolate, and you can find this sort of accumulating in the mutant. So it's accumulating here and is not being put onto the wall, is the conclusions from these experiments. Going on to do some sugar analysis of the purified cell walls. So this is the typical sort of profile you get when you make alditol acetates and you run them through a gas chromatograph. You get a sort of roughly a 3-to-1 ratio of arabinose to galactose. But in the mutant, what you find is the amount of arabinose has gone way down. So this is suggesting this is where we perturb the synthesis of the cell wall. So this is the sort of overall architecture in terms of a flat format of the wall, of the arabinogalactan. So the other useful thing that you can do is permethylation analysis, and this gives you a complete sort of fingerprint of the sugar residues that are present within the polysaccharide. So we have terminal arabinose, 2-linked, 5, T-gal, 3,5-ara, 5-gal, 6, and 5,6. So this represents all the different linkages that we have in the wild type. What you find in the mutant is that the amount of arabinose, as predicted from the alditol acetate analysis, has gone down completely. But what's key is that a residue that we had here, which was 3,5, is completely missing now. So what this suggests to us is that what we actually have now is this sort of truncated cell wall structure. So we've gone from this architecture in the wild type to this now in the mutant. So the mycolates are down in terms of mycolation, which is what you'd expect because we have less termini. We've lost the 3,5 residues and we only have this linear 1,5 backbone. So this has really identified a potential drug target for us in terms of cell wall synthesis, in that if we try to knock out this particular gene in Mycobacterium tuberculosis, we can't. So this sort of summarizes where we're at in terms of arabinogalactan synthesis. This is the galactan backbone. We have AFTA here, adding the first arabinose residue. We have the bulk of the arabinan synthesized by EMB A and B. We have AFTC adding this branching residue. We then have EMB A, B adding again the bulk of the 1,5 residues, and we have AFTB sort of capping arabinan synthesis. What I'd like to do for the last 10 minutes is just talk a little bit about lipoarabinomannan biosynthesis. Again, it contains a arabinan domain which is structurally very similar to AG. It's a major component of the cell wall. It's built on a phosphatidyl-inositol anchor, and its sort of pathway runs in terms of phosphatidyl-inositol monocyte biosynthesis giving lipomannan to give lipoarabinomannan. Again, it starts from PRA, where we have addition of the first mannose residue. We then have acylation. We have addition of a second mannose residue, a third from PRC, a fourth, and then at this point, the biosynthesis changes because at this point, you're using basically sugar nucleotide donors. We have flipping of the substrate, and then we have continuation in terms of synthesis of the mannan backbone through the series of enzymes, MPTB, which again, a bit like the arabinose, but here we've got mannose linked to phosphate and decaprenol as our lipid link donor, where we had the extension of the PIM4. We then have branching occurring here by this enzyme, further extension by MPTA, additional branching by this enzyme to give our mature lipomannan. We then have the addition of the arabinan segment, which occurs through MC. And in some organisms, we have non-capping, such as Mycobacterium coloni. But generally in the sort of slow growers, we also have sort of mannose capping. And in Smegmatis, we have PI caps. So I mentioned MC was what we discovered at the same time as M, A, and B. And this is really involved in addition of the arabinose from DPA. So we sort of have this sort of long-standing interest in sort of lipid-linked sugar donors, and we wanted to understand how DPM was actually synthesized. And it's related to sort of eukaryotic dolichophosphamannose synthases, and through analysis we were able to identify basically the gene, which we referred to as PPM1. It's interesting that in tuberculosis it seems to be a fused gene product. The one half has the catalytic domain, that's the glycosyltransferase, but the other half seems to be involved in a sort of an acylation process. But in Smegmatis and Corynebacteria, actually, this single gene is actually split into two proteins. So again, we sort of, in this case, we knew that this particular gene would be essential, so we generated a strain where we had an inducible copy of a second gene present and then knocked out the chromosomal copy. So what you see on solid agar plates in the presence of anhydride TET is we get sort of colonies forming, but when we deplete the anhydride TET, we get lysis and basically less colonies of the bacteria growing. The two experiments that we can do are shown here in terms of what's going on when we grow the bugs. If we do a 2D analysis of labeled polar lipids from the mutant, so with anhydrous TET you get this normal sort of complex profile of phospholipids in terms of the different sort of monosylated phospholipids that we get in Mycobacterium tuberculosis. But what you find is that when you remove the anhydrous TET, what we see is a loss of the higher PIMs and accumulation of the lower PIMs. And this really shows that we're getting extension predominantly after the PIM2, PIM4, as I mentioned earlier, in terms of synthesis. You can look at the LAM as well and the lipomannan. So in the presence of anhydrous TET, you've got the two products. When you remove the anhydrous TET, what you got is a lot of lipoarabinomannan, but no lipomannan. So that's a bit sort of puzzling. But if you think this is a labeling experiment and you'll have endogenous precursors, what you're getting is a chasing through of your endogenous LM that's already present into your mature lipoarabinomannan. You can make membrane preparations and look at the relative activity of the protein, which is quite easily done. And what we find is that there's a quite stark reduction in terms of activity when you remove the anhydrous TET. The second protein I wanted to talk a little bit about was the MC protein. So one of the big problems is, is that these proteins are difficult to work with because they're full of transmembrane domains. But one of the things that we noticed that they all tend to have this sort of cytosolic C-terminal loop. So we clipped this off and we purified it and solved the crystal structure and also complexed it with a sort of artificial acceptor that we were using at the time. And this is basically a disaccharide of arabinose. And really what we found from the fold was that the C-terminal loops belong to carbohydrate binding module in terms of its fold, and as characteristic of many sort of glycosyltransferases, glycosidases, and receptors. So this gave us a sort of first inroad in terms of the structural basis of one of these glycosyltransferases. One of the other sort of things that it allowed us to do in that we had a mutant in MC, in smegmatis. Basically, if you look at the profiles, you have LAM and LM. In the wild type, in the mutant, you just have LM. If you add back MC, you can get restoration of LAM synthesis. But one of the things the structure allowed us to do was that there was these conserved tryptophan residues, which when you actually made site-directed mutants on the plasmid and put these back in, you didn't get any synthesis of LAM. So it allowed us to explore sort of structure-function activity relationships in terms of the protein and in terms of the product that's synthesized. One of the final sort of points I was going to draw on was that when we discovered AFDC, which was obviously a branching enzyme involved in arabinogalactan synthesis, could it also be involved in branching lipoarabinomannan in terms of the arabinan segments as well? So we started to investigate the sort of synthesis of lipoarabinomannan in our AFTC mutant. And it was interesting that here's a wild-type profile of LM and LAM, and here's our mutant, and you can see the product's shifted, so we're getting a slightly smaller product of LAM in our mutant. So we went on to purify both the lipoarabinomannan and the lipomannan from the mutant. And it was clear that the shift in size was also reflected in terms of the shift in size based on mass spectrometry. And the second technique that we can use is NMR, and basically using 2D NMR, you can obtain a sort of fingerprint of the sort of proton anomeric region and the C13 carbon spectra, which gives a characteristic signature. It's something different from the use of using GCMS and using permethylation analysis. But what it allows you to do is to give a sort of complete fingerprint of the different resonances that we find in lipoarabinomannan. what's key is that basically in the mutant we have the same sort of backbone residues for the mannan segment. But what's highlighted is that we only have alpha-1,5 linked arabinan. We don't have any of the other residues which we associated with the branching or the termination of arabinan synthesis. So this really suggested that we had a very simplified structure in terms of lipoarabinomannan. So one of the things that we thought at the time was that this is probably what it looks like, basically similar to what we had in arabinogalactan, a very short arabinan segment based on its GC analysis and its NMR analysis. You know, how could we use it to probe the role of MC? So one of the things that we did was we did some labeling experiments, and basically you can see here basically LAM, LM in our wild type. In our mutant, we've got our sort of hybrid LAM here, and there's our lipomannan. What you can see here is that when we add in a ethambutyl, we get a shift again in size. So this suggests that basically the ethambutyl that we're adding is actually decreasing the amount of arabinose in our mutant even further. And we know that ethambutyl targets alpha-1,5 arabinosyltransferases. And this can be shown again by if we take out those products and look at them in terms of glycosyl composition just based on hydrolysis. So typically in the wild type, you'd have arabinose mannose like this. In our mutant product, we'd have arabinose and mannose, but there'd be much more mannose because obviously that's the predominant sugar. When we have the sample, when we've treated with ethambutyl, you can see that the amount of mannose has actually increased even further. But what's key is that there's still a small amount of arabinose present. So what this suggests to us is that we're going from this in our mutant to a single arabinose when we treat with ethambutyl. Now, one of the key questions for us now is that are these singular arabinose residues also added by AFTA as we find in arabinogalactan synthesis, or is there a different enzyme involved, which we're currently studying? So this really sort of gives a summary in terms of the biosynthesis of lipoarabinomannan, starting from basically PI. We had the synthesis through to PIM4 through a series of monosylation reactions utilizing GDP mannose containing glycosyltransferases. We have this PIM4 intermediate then being extended using PPM or DPM as we refer to it by a series of mannosyltransferases which belong to the GTC family, and then the addition of arabinose from both MC and AFTC up to now in terms of generating the mature LAM. I think I'm right on time at 10 o'clock, and I'd like to sort of finish by thanking my group who do all the work. This is the current group at the moment. And I'd also like to thank the different funding agencies who supported our work over the years. Thank you.